Yeah, hi everybody. I'm going to tell you a story about ZEB2, which is a gene that we think has been contributing a lot to the evolution of our human brain. But before I do this, I want to acknowledge the people who did the majority of this work. This is on one hand Stefan Roberto, who has been a PhD student in my lab, then Jung-Hoon Lee, who is currently a PhD student in my lab, and Vladimir Jovanovic, who you already heard talking yesterday. So what I find very fascinating is how our brain, how our human brain has increased in size a lot during very recent evolutionary time. So if we look at this plot here, you can see time on the x-axis, and on the y-axis, you can see cranial capacity as a proxy for brain size. And approximately 2 million years ago, starting with Homo habilis and then also Homo erectus, we see a huge increase of the size of our brain. And this happened really absolutely rapidly. Just to put this into perspective, the size of a brain of a modern chimpanzee is approximately the same size of the brain that Australopithecus had. And with this increase in the size and also some morphological changes came a set of human-specific cognitive abilities. Just to mention a few of them, so for instance, our ability for complex communication, which includes like spoken language with a complex grammar, is human-specific. We also acquire knowledge much more quickly, for instance, by learning from others, and we have the ability to maintain this knowledge also over many generations, for instance, by having a written record of the things that we already know. There's also the theory of mind, so that basically states that I have some kind of knowledge, some kind of insight in what you might be thinking or what you might be feeling. We are largely independent from the evolution, and we also have some other social abilities. And taking all of this together made us as a species a very successful species. While we can name these abilities, much less is known about actually the molecular basis for these phenotypes. And what we want to understand is like what genomic changes, what regulatory changes have been contributed to the evolution of the larger human brain and our cognitive abilities. What you can see here, so basically network approaches have been shown to be very suitable approaches for getting some insight about the molecular basis of complex traits and complex phenotypes like the ones I just mentioned. This is a study that we had published already a long time ago, so I'm not going into all of the details, but I want you to know what you're seeing here because this is actually how we found ZEB2, our candidate gene that I'm going to talk about today. So what you can see here is a weighted topological overlap, and the nodes are transcription factors. And these transcription factors are connected with each other, they are linked with each other if they are sharing a lot of target genes, if they share a lot of interactions, and if they share a lot of target genes. So if there's a big functional overlap in these transcription factors. And we derived this network by looking at genome-wide expression data from human brains. We also looked at the same network in the chimpanzee using chimpanzee brain expression data. And basically what you can see if you compare these two networks, that overall the structure or the topology of these networks is very similar. But what we were actually interested in is to see if there are some transcription factors that show some evolutionary differences in the way they are integrated. And especially we wanted to see if there are some transcription factors that would have more links in humans or in chimpanzees. And this is what you can see on this plot here. So basically on the y-axis, you have the difference in the number of species-specific links for all of these transcription factors that I was just showing you in this network. And you can see them on the top if they have more human-specific links, and you can see them at the bottom if they have more chimpanzee-specific links. And this would be 2 standard deviations. So you can see that there are clearly some outliers, and the top 3 outliers, or the top 3 transcription factor with having more human-specific links is ZEB2. And we looked at ZEB2 a little bit more closely, first by starting by looking at the literature, and that got us really excited because from the literature, it was already known that this is a transcription factor that is important for the development of the nervous system, and mutations in ZEB2 have been associated with Mowat-Wilson syndrome. And patients who are suffering from Mowat-Wilson syndrome, they have a smaller brain, and they also suffer from intellectual disability. So that means that they have IQ levels that are way below the average IQ level that you would find in humans. So that tells us that ZEB2 is important for, on the one hand, the development, but also the maintenance of a normal, healthy brain. So any evolutionary differences that we would find in ZEB2 might actually tell us something about the evolution of our brain. So we started working on ZEB2, and while we were doing this, this paper was published. This comes from the Lancaster group. And what they were doing, so they actually used induced pluripotent stem cells from humans and from gorillas. And they initiated neuronal differentiation in these iPS cells. So basically, they let them develop into neurons and then also into organoids resembling brains. And at some point during this neuronal development, the progenitor cells, which you can still see here, they start to change in shape, so they get much longer and they start to detach from the lamina. And while they are doing this, they transform into epithelial cells, and then they start to become neurons. And what this group showed was that ZEB2 is important for actually making this transition to neuroepithelial cells. And interestingly, this step is delayed in humans. So ZEB2 expression comes up at later time points during human development compared to the gorilla. That means that in humans, our progenitor cells have more cells to undergo more cell divisions. And in the end, that would give us more neurons, and that is contributing to the larger human brain that we have. So taking all of that together, we really thought this is a gene that we have to investigate a little bit more closely. So we were asking what genes are regulated by this transcription factor ZEB2 in different great ape species, and can we see actually differences in their target genes between different species? And might these functional changes link to the evolution of the human brain? For our study, we used B lymphoblastoid cell lines. The reason for using these cell lines was that we can get them from multiple individuals. And that was something that was very important to us because we really wanted to have biological replicates. So we were using 3 different human individuals, 3 different chimpanzee, and 3 different orangutan individuals for that study to really make an inference in the end about species differences. And we performed chromatin immunoprecipitation studies using a ZEB2 antibody in order to see where in the genome ZEB2 is binding. And we also performed knockdown studies using siRNA molecules. And then we were asking which genes are changing in expression when we are knocking down ZEB2 in these cells. So let's first talk about the ChIP-seq results. So we performed a few sanity checks. So for instance, we looked at the location of our peaks of the binding events of ZEB2, and we could show that about 50% of the peaks are located close to genes. We also looked at the motifs that we would find in these peaks. And we saw that many of the peaks in all of the 3 species that we were investigating had actually the known ZEB2 binding motif. So the binding motif for ZEB2 was known, so we actually could look at this in our peaks. But then we looked a little bit more closely at the motifs that we would find in these peaks. So ZEB2 is binding to a so-called E-box, and the motif that you can see here belongs to this E-box family. But the middle positions that you can see as CC here, these are actually variable between different E-box motifs. And we are plotting them here on the x-axis. So these middle positions can have 2 different nucleotides. And then what you can see here is the percentage. So how many of the peaks that we had would actually have one of these particular E-box motifs. And you can see that there are clearly some differences between the species. Interesting might be this motif here. So this is the one that we see the most often among our human peaks. And so that's something that we want to follow up to really see which genes are linked to this particular motif. Also, when we then associated the genes to these peaks, then we could see some overlap, of course, in the potential target genes, but we could also see some species-specific target genes, among them the human-specific target genes. Many of them are involved in chromatin organization and remodeling, and I think that also makes a lot of sense for a transcription factor that is involved in developmental processes. For the knockdown experiments, we used 2 different siRNAs to knock down the expression of ZEB2 in our cells. And then we first looked for each individual cell line, so basically for each individual, which genes have been changed in expression due to this knockdown. And afterwards, we were asking which genes actually show a species difference in the expression change that we got through this knockdown. And this is what you can see here. So all of the lines are representing different genes, and they are shown in reddish colors if they are upregulated due to the knockdown, and they are shown in blue if they are downregulated due to the knockdown. And I think you can see that there are some species differences. So the first columns are humans, then we have the chimpanzees, and then the orangutans. So we have a couple of genes that seem to be much more strongly upregulated in our orangutan cells, for instance, but also some that are specific to humans, or some that are specifically upregulated in the chimpanzee cells that we have. So we can now make these pairwise comparisons between species, and we see that the difference, if we compare humans to orangutans and if we compare chimpanzees to orangutans, then we see more differences than if we compare humans to chimpanzees, and that makes sense due to the phylogenetic relationship. I think the most interesting set of genes are these 123 genes that have changed in a human-specific manner. And for these, we looked at gene ontology enrichment, and we were very amazed when we saw that actually the top enriched category among the specifically upregulated human genes is called negative regulation of neuronal precursor cell proliferation. So even though we actually had done our experiments with B lymphoblastoid cells, we got some signature that seems to point to something important going on with neuronal development. So fortunately, we could have a look at these published data set from this other group, and now we could ask in their iPS cells that have been differentiated to neurons for particular time points, what genes are correlated actually with ZEB2 at all of these different time points. And we could compare this between the human and the gorilla that they had done. So first of all, we actually validated that really the expression of ZEB2 is delayed in human compared to gorilla. And then, as I said, we were looking at the correlated genes at these different time points. And what you can see here are the gene ontology groups that are enriched among these correlated genes. And interesting, so if we look at, so many of the genes actually were involved in cell cycle or neuronal development, but again, we see a clear species difference here. So basically, the genes that are correlated with ZEB2, many of them are many of them actually have a function in cell cycle regulation, but only in humans. So this is something that we don't see in the gorilla cells. On the other hand, already from the very early time points on, we see a positive correlation of ZEB2 with genes involved in neuronal development in the gorilla, but this is only coming up in the very end for the human cells. So in summary, I hope I could convince you that ZEB2 is a very strong candidate for having been involved in the evolution of the human brain. And we saw some species differences in their target genes. And it seems like some of the human-specific features in brain development and basically our ability or our cognitive ability, some of these seem to be linked to differences in target genes of ZEB2. And with this, I want to acknowledge many of the collaborators, also other group members that have definitely contributed in many of the discussions in the group meetings to this project, and also the funding agencies. And I want to thank you for your attention.
Are there any questions? I also encourage you to use the app, the online app. We have time for one question. You, please.
One question.
Hi. Oh, yeah. That's all right. What do you think could be an environmental factor that allows these molecules, ZEB2, to produce in the evolution of human that exponential growing of human brain in the last in the recent millions of years. What do you think it could be that factor that allow this exponential growing related with these molecules of this ZEB2?
So basically, ZEB2 is important for during this neuronal development. So maybe I can go back to this figure. So you were asking about ZEB2, what the role of ZEB2 is in making the bigger brain, right? Yeah. So on the one hand, what we know is that the expression of ZEB2 is coming up later in humans compared to gorilla. And ZEB2 seems to be at least one of the most important molecules for pushing the transition of these progenitor cells into the epithelial cells that would then later become the neurons. And because ZEB2 is coming up later in humans, the human progenitor cells, they can undergo more cell cycles, so they can undergo more divisions. So basically humans are producing more progenitors because they have more time. And then we have already more progenitors, and all of these progenitors would later on become more neurons, basically. And so that would be one of the contributions of ZEB2. And then from our work, so basically we were looking at what are the actual genes that ZEB2 is regulating. And there we also see genes that would be involved with cell cycle regulation and with neuronal development. So yeah, it's basically regulating other genes that then would be important for this neuronal maturation.
Thank you.
Yeah, I'll try to make my question a really quick one. So parsimony would say that ZEB2 is where changes might have happened. Have you taken the chimp ZEB2 and put it into the human cell lines and vice versa to see if and then done the chip-seq to see if you get different levels of peaks?
Very good question. So this is something that we actually currently do. So we left the B lymphoblastoid cells because now we have iPS cells from different primates. So with these iPS cells, we have a chance to now go to a neuronal context. So we can, as these other guys did, we basically can differentiate them into neurons. And we have iPS cells now from 3 different humans, chimps, and orangutans, actually. And so currently we are doing exactly what you're proposing. So we want to see what the chimpanzee transcription factor is doing in the human cells, but also vice versa, to see if basically with the human ZEB2, we can make the chimpanzee cells a little bit more neuron-like, quote unquote.
Okay, thank you for your